respected Director and Commandant AFMC, dignitaries, esteemed faculty members, distinguished alumni, valued students from all over the nation. It is with immense joy and pride that I extend to you a heartfelt welcome to the 15th edition of Illuminati, the annual undergraduate medical conference of Armed Forces Medical College. What began in 2010 as a visionary initiative has over the past decade and a half evolved into one of the most esteemed and impactful platforms for undergraduate scientific discourse and medical innovation amongst medical colleges. We are deeply honored this year to host over 1,300 participants from 58 premier medical institutes across India to embody the ethos of Illuminati, the spirit of curiosity, knowledge sharing, and lifelong learning. Central to this academic fest is the CISO, the Student Scientific Society of AFMC, whose unwavering dedication, meticulous planning, and execution have consistently elevated this conference to newer heights. This year holds special significance as we mark the inauguration of the UG Research Hall of Fame, Anusandhan, and also two pioneering initiatives, Akarjan, the 3D printing lab, and Anveshan, the tinkering lab, both made fully operational within a few weeks. These state-of-the-art facilities are catalysts for innovation, providing our cadets and young researchers with the tools to transform ideas into impactful solutions. In addition, the clinical quizzes of the Student Scientific Society have further elevated our academic profile by organizing 3 national-level quizzes, namely on tuberculosis, multiple myeloma, and diabetes mellitus, engaging participants from across the country. The active participation of our cadets in prestigious forums such as the International Conference of Military Medical Schools and national platforms like the Indian Myeloma Congress stand as a testament to their academic excellence at both national and international events. We reaffirm our dedication to collaborate, to uphold the highest standards of scholarly pursuit, and remain unwavering in our service to science and to humanity. May the glow of Illuminati continue to guide and inspire all of us. Thank you.
We're beyond thrilled to announce that the L Batch has donated 7.24 lakhs to CISO to sponsor the hackathon events in all the subsequent Illuminatis here at FMC.
We request the
compliment to the team of Illuminati instead of blowing up this.
While we are showcasing our countless achievements, how can we look
the club in print?
packed with articles from cadets and officers alike? It covers everything from scientific milestones to behind-the-scenes chaos. Whether you're into research highlights or just your go-to.
We now request the dignitaries on stage to kindly launch this year's edition.
and a deeply humbling moment for me to stand before you today to deliver the vote of thanks as the Secretary of the Student Scientific Society and the Organizing Secretary of Illuminati 2025, AFMC's annual National Undergraduate Medical Research Conference. Illuminati 2025 is a dream months in the making, and today's inauguration marks a significant milestone in that journey. At the outset, I would like to express our deepest gratitude to our Director and Commandant, Lieutenant General Pankaj P. Rao, whose presence and guidance has propelled our conference to new heights. I would also like to thank our Dean and Deputy Commandant, Major General Kiriraj Singh. Sir, your unwavering belief in the caliber of your cadets has been instrumental in making Illuminati 2025 a reality. A very special vote of thanks goes to Brigadier Suman Kumar and the Officer in Charge, Student Scientific Society, Colonel Vayudev. Sir, words fall short in capturing the depth of our gratitude. You stood by us through every challenge, offered reassurance every time we faltered, and pushed us to aim higher at every step. I also extend a sincere and heartfelt gratitude to all our HODs and our esteemed faculty members. Your guidance, encouragement, and presence here today have added tremendous value to the conference. Your mentorship forms the foundation upon which we grow as young academicians. I would also like to sincerely thank Brigadier in Charge Adam, Commodore Adam, Colonel Training, the PMC of the Officers Mess, the Quartermaster, and the Company Commander for facilitating numerous administrative and logistical processes behind the scenes. I would also like to thank the Headquarters Southern Command and all its affiliates for facilitating the Join the AFMS campaign. A special word of thanks to all the tech support staff, maintenance teams, and support personnel who worked tirelessly behind the scenes and ensured that every venue was clean, every setup was in place, and every request was met with a smile. To my incredible H3 batchmates, especially the core committee, thank you for pouring your heart and soul into every detail. Your grit, efficiency, and teamwork have been the heartbeat of this conference, and I am truly honored to have worked with you all. To our junior batches, your enthusiasm, energy, and eagerness to help have been heartwarming. You've carried out your responsibilities with such diligence that has made us so proud. I would also like to thank the press and media for taking time out from their busy schedules to cover this confluence of youth and medical research. Finally, to all the 1,342 delegates from various medical colleges across the country who will be attending this conference, thank you for being a part of this moment. With that, I invite you all to immerse yourselves in the scientific spirit of Illuminati 2025, igniting prodigious minds. Let the next 2 days be a celebration of innovation, intellect, and inspiration. Thank you, and Jai Hind.
Hello.
So myself, Brigadier Suman Kumar and Major General P.K. Sharma are deeply honored to be sitting here chairing this keynote address, and it's my singular honor to introduce Dr. Debojyoti Chakravarti to all of you. Dr. Debojyoti Chakravarti is a leading expert in genome editing and RNA biology, currently serving as a principal scientist at CSIR Institute of Genomics and Integrative Biology. He earned his Masters in Molecular Biology from West Bengal University of Technology, Kolkata, and a PhD from Max Planck Institute of Molecular Cell Biology and Genetics in Dresden, Germany, under Frank Buchholz. After postdoctoral research at TU Dresden, he established his lab at IGIB, focusing on embryonic stem cell biology and CRISPR diagnostic. His work includes developing Feluda assay, a CRISPR-based tool for detecting genetic disorders like hemoglobinopathies, showcasing his clinical potential. Dr. Chakravarti's research also explores therapeutic genome editing for genetic disorders and stem cell-derived organoid models for disease modeling. He has received the CSIR Young Scientist Award 2020 and is an EMBO Young Investigator, reflecting his scientific excellence. Beyond science, Dr. Chakravarti is an accomplished sitarist, performing widely in India and abroad by blending art and research. His keynote on CRISPR-Cas in medicine will inspire the medical MBBS students who are here, highlighting its transformative potential in healthcare. I request Dr. Chakravarti to please come on the stage and deliver his address.
So good morning everyone, while this gets loaded. So first of all, it's an absolute pleasure to be here, all the dignitaries who are present here. My only connection with AFMC is a failed medical entrance exam. Okay. That's almost 20 years back or so. And at that point of time, I just wanted to visit this place and see how this is, because we used to grow up with knowing about this great medical colleges of India, and AFMC is right there at the top. And of course, it didn't make it, but and that's why I went into the natural sciences, the BSc, MSc, PhD. But I'm just glad that I finally got a chance to come here. Working with a lot of clinicians, this has been a very special experience actually to come here and see a different setup rooted in so much in service, sacrifice, valor, and of course excellence in medical sciences. So I'm really grateful that I get this opportunity today. So thank you sir for the very kind introduction. In case you don't understand the CRISPR talk, there's an easier talk which will be after the break, so you should not go. Okay. So I'll talk a little bit about some of the work in gene editing, which we are trying to do over here. I'm told that most of you are undergrad students. So with a quick show of hands to help me understand, how many of you have heard and know about gene editing and CRISPR? Okay, I only see at the back, so which means I have to explain a little bit what this technology is about before going into details. Okay. So I come from an institute in New Delhi, which is called Institute of Genomics and Integrative Biology. And this institute was, it was entrusted with sequencing the first Indian human genome, and that they completed in the early 2000s and so on. And then also went on to do the human genome sequencing of other neighboring countries, Bhutan, Malaysia, Sri Lanka and all. And we got a lot of sequence information. So for the quiz enthusiasts, the number of letters are in high billions, the ATGC letters in the human DNA. So once they knew what are the alphabet in the Indian human DNA, or for that matter, human DNA, they also found out that there are many mistakes in that. So the technology that I'm talking to you about is about correcting these mistakes. And for a long period of time, it was felt that you are born with whatever is there in your DNA. You must have heard that DNA is the blueprint of life and so on and so forth. And there is no way to change this once you are born. How would you change that? But thanks to some of these almost technologies which has really changed the paradigm of how we look at biology and diseases and therapies, it is now even possible to do these kind of changes, and it's not a holy grail anymore. So much to the extent that we have in the world right now people like you and me who are walking about with their DNA altered. So that's the power of gene editing per se. So what we are trying to do here is to correct errors in the DNA, and I will tell you how this is being done. And I'll also tell you a little bit today about the talk that I'm going to give. It's not just about the science per se. The science is, of course, great, and you keep on improving things. But sometimes we have to think beyond what we really develop in the lab and think of how we can make it possible for patients to have it. You're all, doctors and, going to become doctors. As long as science is remaining fixed in the lab and there's no way to translate it, sometimes it loses its stigma, sometimes it loses its power kind of. So how to make these things affordable, accessible for people across the country and the world. So that's also something that I'm going to talk about today. So, like I said, that it is no more a holy grail. There are patients who have been treated with, for CRISPR therapies, gene editing therapies, for a disease that you guys have studied. I have seen some sickle cell anemia camps in the first part of the presentation. So sickle cell anemia is a disease which has actually been cured. When I say cured, it's a very big word in medical, because you technically cannot be cured, just your symptoms get away. But here the genetic basis of the disease has been obliterated, which means that the patients are actually fine. They do not have any, let's say, remnants of the disease. So there are patients who have been cured in the US, but then this got approval only in 2023-24. It's not a very early, long period back. But as you see in this article in Nature, I've highlighted here, this one-time treatment is very costly. You have an idea about how costly this treatment? It's a one-time because you're changing your DNA, so essentially you're cured for life. But this treatment costs close to about $3 million per person. So that's about 20 CR, okay, 20 crores to pay to be free of a disease. Okay. Now imagine, where do the largest number of sickle cell patients in the world come from? They come from Africa, and followed by India, actually. So if you had to make such a life-saving treatment possible, our patients will never be able to afford that. And you will be very surprised, maybe you know this already, that most of the sickle cell patients in India, they come from tribal belts. Really the poorest of the poor people. Maharashtra is a huge sickle cell belt also, for example. So therefore, the job that I am trying to do here in IGIB is to see that not only make such therapies possible, but also how to make it accessible. And that's a very complicated puzzle. Really, science is very easy to do in the lab and make therapies, but to make it available to people, that's a completely different domain. It's more complicated than getting the Nobel Prize probably, because there's a lot of different areas how you have to tackle that. And I will tell you also why this is so costly. So these two individuals, you might know them. One of them definitely is Jennifer Doudna on the left. She won the Nobel Prize, for gene editing in 2020. It's a fantastic, scientist from UC Berkeley. And Fang Zhang, who is from MIT. Now, why I mentioned this, because they were, they were fighting for a long time to have the patent rights over CRISPR. When you have such a path-breaking discovery, you want to own that. And UC Berkeley and MIT had been fighting over this for a long period of time. Who owns the access and who owns the royalties essentially to get CRISPR for CRISPR, which means that anytime anybody in the world uses CRISPR for some application, they will profit from it. And what happened was that through a lot of litigation and legal issues, finally Broad MIT actually got the patent for CRISPR just a couple of years back. Now that's okay, because as you might know, except for those landmark things in insulin where patent rights were waived off and things like that, most of the medical technologies or developments are actually patented, because it also provides a framework for protecting your intellectual rights and so on. So it would have been nice had this been as simple as that and one could actually start therapies elsewhere. But if you look at how the Broad Institute MIT tries to give these rights for making therapies, it's through a very complicated process. So if you go to their website, you will see actually how they make this available for different countries, different patients and so on. And this is through this very complicated network where they have their own companies. And if you, supposing today you want to make a gene therapy for, let's say, breast cancer, BRCA1, you want to correct the mutation and make a therapy like that, and you have a company you want to set up to make these drugs, you would actually have to go through these companies which have been set up in the West, and you pay them millions of dollars of royalty or millions of dollars of license fees till you get actually the possibility of even making these therapies. Okay. Now that's not possible to do for resource limited settings, or for that matter, even in India, a company which will not have very deep pockets will not invest in such kind of drugs, and that's why these drugs will forever stay away. Even in India, for that matter, anywhere in the world, except where there is really the muscle power and the economic power to do that. And there's no way to get through it, because that's how these patent laws are built, that's how the universities profit. And this means that even though these therapies are available, FDA has approved them, they will never be available for the patients who need them the most. So if I could summarize what we need to make this available, one of course is the therapy safety, that how safe you make these technologies. We are correcting an error in the DNA. So what does that entail? The pricing has to be regulated in some way. How you can do that regulation? Of course, a very important part is the IP and licensing, intellectual property. How do you manage to generate intellectual property yourself? And then, awareness like what I'm doing over here, and also with patients and doctors, that are they ready for such kind of novel technologies and therapies to actually implement. So I'll come to each of them and how we are trying to mitigate here in India and try to see if these are possible to do. So to cut a long story short, this whole technology works on the principle that there is a bacterial enzyme called Cas protein, or a Cas9 protein in this case. This is shown here, and it takes in an RNA as a guide, okay, which is also called a guide RNA, and it can be directed to go to any 20 to 21 nucleotide sequence in the DNA of a cell by putting in information in that RNA that you have to go and bind to that part of the DNA where that sequence actually matches. Once it goes to that site in the DNA, out of 3.8 billion letters, it goes and finds that site. This Cas9 initiates a double-strand break in the DNA, and then once the DNA is cut, what happens when the DNA is broken? It is repaired. Because by the time that I started this talk and till here, I would have had 1 million DNA breaks in my cells, which has gotten repaired. Our cells is very, very efficient in repairing. So it repairs that. Now scientists are smart. What they did is that they saw if you put in a small piece of foreign DNA at that site where the DNA double-strand break has happened, you can actually integrate that. Now imagine if there was a BRCA1 mutation, you cut it at the BRCA1 site using this kind of a technology, and then you put in a foreign DNA which is the correct nucleotide sequence, you can essentially gene correct what was originally a mutation into a normal cell. Now that's a very simplified way of telling all these things. If you have to take it to therapy, it's a very complicated process. Where would you do it? How would you do it? And so on. But what we saw was that this Cas9 that was being used all over the world, it comes from a bacteria called Streptococcus pyogenes. So it's a bacterial defense system which has been harbored for using in human cells. So the Cas9 originates in the bacteria. It was used as a defense mechanism against invading bacteriophage or virus. And we have managed to take this Cas9 and put it in human cells and do the same activity and essentially change the DNA sequence. So when we were working on this, alternative Cas9, so most of the work that I do here is with this gentleman here, Dr. Shobik Maiti, who is currently the director of IGIB. He's a friend, mentor, and a colleague. He comes from nucleic acid biophysics background. Okay. So what we found out that if you take in a piece of DNA like this, and then you incubate that with the Cas protein, what happens is that it cuts, it's an endonuclease, so it cuts the DNA. It makes two bands like this because it will have a bigger band and a smaller band. It cuts in the middle. If you do that with Streptococcus pyogenes Cas9, or if you do that with this novel Cas9 called Francisella novicida Cas9, which is from a different bacteria which we are studying, it always does this cut. But interestingly, whenever you take another piece of DNA which has got some mismatches or a mutation in them, let's say 2 mutations, 3 mutations or whatever, whereas Streptococcus pyogenes Cas9 would cut these as well, we call these as off targets in the DNA because they are really off targets, okay? FN Cas9 would not cut these. Now that's very interesting because that tells us about an important thing called specificity. Where your drug molecule should only act on the substrate that it's supposed to act and not on something else which is very similar. That's called a specificity. And why is specificity important? Because it ultimately leads to safety of the drug molecule if you put it inside the human body. You only want to cut the BRCA1, you do not want to cut P53, right? So that would be a problem if that happened. So we thought, okay, that's very interesting because, this Cas9 has actually entered clinical trials, whereas the Cas9 that we are working with, it seems to be very, very specific in terms of what it is doing. And we realized, you don't have to go into details about these graphs and all, but just to suffice to say that we did a lot of protein DNA binding assays to understand that this gas is so specific because it can, whenever it sees a mismatch, with the RNA and the DNA, it evicts from its off target. It doesn't bind to the off target anymore. It just comes off. But if it finds a complete match, then it sits there and it cuts the DNA. So that is essentially being told over here using binding affinities and so on. So that was something interesting because we thought that if you give this piece of Cas9 a piece of like a DNA which has got full match with the intended sequence, and you give this another piece of DNA which has got a single mismatch, then this Cas9 should be able to tell what is a full match and what is a mismatch, right? Because it has so much of sensitivity towards a single mutation. So if we could convert all that into a kind of a readout, then we could sequence a mismatched DNA without the need for sequencing per se. You can use Cas9 to do that. So if you remember, when you do sequencing, DNA sequencing like Sanger, NGS, you're actually reading the DNA and you're telling if there is a mutation in that. And doctors often use that to understand if the patient is suffering from a disease, rare disease or genetic disease and so on. So this was possible, and this is actually the origin of this technology which we call as Feluda. Okay. So before I go on, are there any Bengalis in the group here? Yes. Okay, there are 4 or 5. So you might be knowing who Feluda is and what that it is. Just on 2nd of May was the birthday of Satyajit Ray. Satyajit Ray is a big icon in Bengal. And Feluda happens to be a detective character which he built, made in movies and stories and so on. Feluda actually is the world's first CRISPR-based paper strip test for detecting point mutations. Okay. I told you why it was made that way, because the Cas9 can do that detection and so on. And why we kept the name Feluda was because during COVID times, when all research was stopped and the government said you make some kind of a detection for COVID and all, we were kind of competing with 2 other groups, and those 2 groups are the ones that I showed in the beginning, Jennifer Doudna's group and Feng Zhan's group, and they gave names, one based on Cas13 called as Sherlock, and the other one based on Cas12, they called it Detector. So we wanted to give it a Desi detective's name, okay? And since we were both Bengalis working in the team, we gave it Feluda. That was the origin of the Feluda. And we conceptualized this in somewhere in January 2020, and this got approval from the drug controller in September. So it's just about 6 months, 7 months to develop this test. Essentially, I won't go into details, but we married a little bit of CRISPR biology and paper strip chemistry. We made it like a paper strip test completely, where like a pregnancy test, essentially, where you can detect through this appearance of these bands whether a sample is positive for COVID or a positive for, or negative for COVID and so on. And these were, things that we could develop to help the clinicians to understand whether this test is positive or not. Tokshan, Jatayu, or characters from Feluda stories and so on. But really, we actually made this not for COVID. It was some kind of a thing that came out separately. We actually made it for sickle cell anemia, because that's where a point mutation has to be detected quickly on the field without needing these samples to go to a Sanger sequencing. And sometimes our patients told us that they coming from tribal beds, they would come give their blood like you see over here. And then it goes to, let's say, the nearest Sanger sequencing thing, which is in CCMB Hyderabad, for example, and then it will take almost a month for the results to come in. By that time, the patient would have gone. So we wanted something where these kids could simply, spit into a small tube. The DNA will come out, molecular biology techniques to break the cell. DNA comes out, you do a CRISPR test, and within 2 hours, you will know what a Sanger sequence can do. So that's what we had developed, but then, we had to make it for COVID, and then, if you can play this movie. Yeah, so of course we are an academic institute, so we had to partner, we had to do the technology transfer. So we transferred this to, I think this has gotten probably once more. Yeah, we transferred it to the Tatas, the House of Tatas. They made a new company called Tata Medical and Diagnostics. They ramped up this whole thing into like a robotic system, put it behind trucks, and then they went to all the places where qPCR machines were not available during the pandemic and so on. And that's what they did over there. That's all fine, but it's more like a distraction because you had nothing else to do during COVID, so we had to put everything in the diagnostics. The ideal place would be to see if we could do gene editing with high specificity. Of course, Feluda is not dependent upon the disease. You can take any other disease. This is, for people in the gastroenterology department. This is work that we did with Dr. Govind Makharia at AIIMS, where he said that in our OPD, people come with, clarithromycin resistance phenotypes, which we cannot do without doing a 5-day culture of these kind of samples. So you could do this using Feluda also, just quickly within an hour or two, to detect if this bacteria is present or not. This came from the northeast, where in Arunachal Pradesh and so on, where rotavirus is very prevalent. And again, there also in the pediatric department, they do not have mechanisms by doing this kind of very quickly without having to culture this bacteria in plates, 3 days, 4 days, and so on. So we made these kind of tools to actually do these tests using CRISPR very, very quickly with a quick turnaround time. But like I said, that the main approach is not to make it for diagnostics. If you have something which is so specifically binding to target DNA, ideally you should be able to cure a disease with that, make corrections, genetic corrections. So cut a long story short, what we did then is that we had a wild type Francisella novicida Cas9, which was quite efficient, quite specific, but it was not very, very robust in doing the gene editing at the site where it was supposed to do. It's a wild type enzyme, okay? So we collaborated with a Japanese group, and we figured out that if you make different mutations on this protein, so in a protein there are amino acids, right? If you change amino acids, then you change the properties of how this enzyme can interact with DNA. So if you make very directed changes in these amino acids, you can actually make the wild type enzyme perform many, many times better than actually so we call this as engineered Cas proteins. So that's what we did. So it now became very efficient, and it was even more efficient than the ones that have gone into clinical trials, Streptococcus pyogenes Cas9, without losing this high specificity inside cells, even towards one mismatch of targets. Okay, so one base pair mismatch in the genome out of 3 billion letters, it is still able to distinguish that because of the specificity. And we could show this in different types of locus and so on. Strikingly enough, if we take one of the most common locus here, which is for sickle cell anemia, and you look at all the chromosomes where this can go and bind, if you compare it with the one that went into the FDA approved trial, you would have close to 1300 off targets across the chromosomes. Now this is very dangerous because if a Cas goes and binds and cuts to other chromosomes, it means that this might lead to chromosomal breaks. And sooner or later, there could be some chances of having some kind of a chromosomal break related problems, which could end up in having a cancer phenotype. What we saw was that with these engineered Cas proteins, we were having almost undetectable off-targeting, except for that on-target here, which is the HBB gene, which has the sickle cell mutation, for example. And we validated these in patient samples, and we saw that it was very, very specific. So that was something that is very, very unique. And only because of that, we could actually make, second generation, third generation CRISPR-Cas systems. I will just show you one example of that, where this is work done with the LV Prasad Eye Institute, and Dr. Indu there came and said that we have a patient here who is completely blind, 29 years old, and that happens because this patient has got a single point mutation here, this TAG, which is the A, which is a mutation, and that caused this disease called Leber's congenital amaurosis. Okay, and is it possible to correct that mutation back to normal? So we made what is called as induced pluripotent stem cells from the patient's cells. So we take the fibroblast, skin fibroblast, convert it into stem cells, and then converted the stem cells into the retinal cells, okay, by differentiation. And when you do that and you do a gene correction in there, this is a very beautiful thing to see that a protein which got lost from childhood is actually restored back to normal. So this means that if you give these cells this retinal patch and put it behind the cornea, this is exactly what these guys are trying to do now through a trial, you could restore vision. Typically, these things are done by finding a matched donor, and make that and putting it behind. But then what happens with match donor, as most of our clinicians know, is that you need to be very, very careful about immunosuppression and the GVHD and things like that. What we are talking about here is your own cells gene corrected and put back to you. So the GVHD doesn't exist anymore, okay? Or to a very, very minimal extent. So this is something that they are taking ahead for a clinical trial as a possible route to correct blindness, for example. And of course, once we had all this, this gave us a very unique opportunity. We got an American patent on it because what we realized, and I showed you the first point, was the IP and licensing. If we keep on relying on technologies that we have to import from outside, then we will have to pay all these licensing fees and regulatory fees. Unless we have our own IP from India, the companies that work in this space will not have the flexibility and the freedom to operate, or the FTO as they call it, to start making these drugs. So this was something that we were able to solve using this R&D. And like I said, that my only connection with the armed forces was because when I was young, we used to watch this in Doordarshan during the Republic Day, have these fighter planes go above and see this in the television. But through science, I would say, we got an opportunity to be there as a guest at the Kartavya Path last year and see this live, and it's a tremendous experience to see that as a guest from the government. But that's all fine, you can make patents, you can sell technologies, that doesn't solve the problem of what we're trying to do, which is to make gene therapies possible in India. So that's a different science altogether, and I will come to that now. And like I say, that the path to the clinic is very complicated. You can keep on doing your R&D all your life, you can write papers, publish great papers and all. But the moment you have to take this and put it really in a patient, or to go through the hurdles that are there, regulatory and otherwise, you will realize that what works in the West doesn't necessarily work over here. It's something that's very, very important to understand. You have this path-breaking technologies curing people, but the moment you try to take it and adapt over here, you realize that the Indian system is so diverse, so complex, you simply cannot do that. And sometimes you have to make these kind of adjustments, where the patient is, it's like a traveling paradox, right? You're inside the train and outside the train at the same point of time. Okay, you're traveling regardless. So we have to solve those issues as well. So sickle cell disease is a very important disease from the context of the government. One, because it is, they are rampant across all the countries. Maharashtra is not very, colored over here, but I tell you, it's a huge belt of sickle cell. Most of my calls actually come from doctors. Sorry. They're largely because you're dealing with patients who are coming, like I said, from tribal areas, really very poor people, and the standard line of treatment, as most hematologists know in sickle cell, is to give hydroxyurea. Okay, hydroxyurea is also not available to most of these patients. So imagine telling them about a gene therapy which is going to cause gene correction and cure the disease. So that's almost like a blue sky kind of a solution for this disease. We'll wait for this to come. Sure. Okay, thank you very much. Yeah, so to understand more about how the disease context is, I would encourage you to read this very nice article that came in CNN last year, where the reporters actually went with us to the villages to understand how this has been developed. But essentially to tell you that this was a problem which was picked up by the government since 2016, and they said that this will become a mission in the country. And as you might know, that in a couple of years back, our Honorable Prime Minister actually launched this sickle cell mission as with the idea of eliminating the disease by 2047. So we know that it probably won't be eliminated, but we'll make strides towards how to reduce the burden of it. And it is actually a big problem. Doctors often say, hematologists say that the Indian sickle is a mild phenotype, without in many cases realizing that it's mild because the organ damage is not actually monitored. So therefore, when you ask them that what is the the age of the oldest patient, it is always in the 40s, because these people, they just die at some point. So now, and there are also other complicated things like they have high fetal hemoglobin, yet they present with a vaso-occlusive crisis and so on and so forth. Now, what we did is we tried to see if you can use this CRISPR technology that we developed to actually do this gene correction. And through many years, we showed that this is really possible. If you take an apheresis product from a patient, and then you make this CRISPR complexes to correct the mutation, you can almost get from 80 to 90% of the gene corrections. In sickle cell, there's this GTG to GAG mutation. It's a point mutation. So if you revert this mutation back to normal, you can essentially correct the disease. So we could do that without causing any kind of defects in other, in what is called as an indels in the DNA, which means that no other DNA breaks are happening. Quite safely, they can be differentiated into the reticulocytes and so on without off-targets in the genome. And then these can also go into preclinical studies where mouse models are engrafted and then they can be cured. But that's quite good. These are very possible to do within the lab. The question is it possible to actually take this to the clinic? And something very strange happened in 2022 when the government actually came and said that, why don't we try a clinical trial for CRISPR gene editing in the country, which will be entirely federally funded. Now, typically such a thing doesn't happen across the world because such clinical trials are extremely expensive. They run in hundreds of crores or something like that as a cost, and they are taken up by companies, by investors, and in the West, that's how it is typically done. But nobody would come and, a company would come and do this for tribal patients because there is no return of investment. So the government said that, okay, we are going to fund you for this, whatever is required, but can you set this up in India? So we did that, and we call it as Birsa 101. Birsa, of course, is in honor of Birsa Munda, who is the tribal, superhero kind of, with the idea that we should be able to gene correct cells from the tribal patients or sickle cell patients and give it back to the patient. These would be hematopoietic stem cells, which will be gene corrected and put back into the body after a round of myeloablation. Of course, when you do such a thing, this is it's not the it's way beyond my pay grade when you have so many different secretaries who have to come together to support that. Because you have a DST which funds, you have a Ministry of Tribal Affairs which looks after the people in the tribal community, you have the ICMR which makes the ethics committees and the national ethics committees for trials, you have the DBT which gives the RCGM approval, you have the CDSCO DCGI which gives you the approval to test a new drug and a clinical trial, and CSIR of course, which is my parent thing. So it was only possible because at some point Niti Aayog said and came and said that okay, we are going to do all this for you. So now Niti Aayog monitors it and they bring all the secretaries to the same table, and then we can figure out how to go for the next step. So this has been a truly kind of a collaborative effort, I would say, across all the possible departments that could be involved in this. And to do that, we had to actually set up a completely new GMP facility. Because imagine you have a patient's blood coming in and you have to take CD34 hematopoietic stem cells out of that, do a gene correction, and do this all aseptically without having any kind of contamination, so that the patient who gets enrolled in AIIMS, and AIIMS had to build a ward for these patients there, and they have to get this with that high safety, that there is no contamination during this whole process, and then the patient has to be followed up for any kind of crisis and engraftment. So all this had to be set up. We are currently at the preclinical phase, finishing across the regulatory, and if everything goes fine, perhaps in the next year we might have the first patient dosed. So the last part of the talk, I can talk about details more later on because the time is running out. The last part of this is to tell you that CRISPR or gene editing technologies is disease agnostic, because it's essentially correcting a genetic defect. So a large number of genetic diseases fall under the rare disease category, where you have no possible cure because they are point mutations or mistakes in the DNA, which there are no drugs for. So, and several of them come in the rare category, and in a country like India, a rare is not a rare because of the population size. There are thousands and thousands of patients who suffer from them. So I'm going to tell you a story about such a rare disease patient. So there was this couple who came to us in just after COVID in 22. They said that their daughter was suffering from this ultra rare disease. So within rare, there is a category called ultra rare, which has extremely few individuals across the world. And the mutation that was presented here is this G here is getting converted to A in the SERPINE protein. And what happens when that mutation happens is that the protein tends to aggregate in the neurons. And because of this aggregation, there are seizures, there are epileptic seizures, slowly leading to loss of all sensory perceptions, whether it is vision or smell or anything. And then the patient dies in their teens. So this was a 17, 18-year-old girl who presented with this kind of disorder. And there is no cure, absolutely no cure for this at this point of time. So this parents, they were quite well-to-do. They were funding New York University Langone Center, which is a very top center for schizophrenia and other diseases, and to develop, because they know that there is a gene therapy in the horizon. You could correct this kind of genetic mutation. And they were funding this, but they said that if you try to do this in the US, the regulatory process is so complicated that you're not going to get a drug before 6 to 7 years. And neither is the cost going to be possible to be supported by just the family's savings, for example. So then they came and met us and said that would you be able to take this up? So quite naive as we were, we took this up. I won't do that again, but essentially we had to develop a CRISPR therapy from scratch to correct this mutation, okay? And we did that. So we made a CRISPR, proteins which would actually correct this mutation back to the normal in 80% or so of the cells, which was clinically that's enough. So you don't need to correct more than that. But more importantly, we took this patient's cells, the saliva, because she was already drooling when she came, so we could not take blood from her. So we took that saliva and we converted it into stem cells. There is something called reprogramming, as you might know, also won a Nobel Prize. Through that, you can take any body cell and convert it to a stem cell, like the embryonic stem cells, without having to go to the embryo. So we reprogrammed that, and we differentiated that into neurons. So we made neurons on the dish with that, and we saw that upon gene correction, you could actually reduce this, aggregation back to almost like normal levels. So pathophysiologically also, there was a benefit you could associate. We did the whole genome sequencing of this patient to see if there are any off targets that this CRISPR therapy might have. We found there was only one off target here, but it was lying in a gene desert, meaning there's no gene over there. So you would not hit something by mistake and cause some problem. So all that is good because you have a CRISPR therapy which can correct it. But then you have to put this in the neurons, right? So you cannot simply take a Cas9 protein and put it in the neuron. So there are technologies which are based on viruses. And some of these viruses are called adeno-associated viruses, which are quite, let's say, partially safe for gene therapy because they have been used for gene therapy. These viruses can package this kind of a CRISPR material and can be injected into the body of an individual. And these viruses can be pseudotyped, meaning that their capsid proteins can be changed so that they only go to neurons and not to the liver, to the heart, or so on and so forth. So we had to package all this in a virus, and I'm not a virologist, so I collaborated with one of the top virologists in the country who's making these adeno-associated viral vectors in Bangalore at the Narayana Nethralaya Center. And we put all these CRISPR proteins and we tried out different ways of injecting into the brain in mice. So close to 200, 300 mice in different routes, ventricular, intrathecal, intravenous, found out what would be the immune responses to each of these processes, how much of gene correction we would get, and so on. So all this has to be done if you have to go for a therapy, okay? For a, to see if this is kind of safe and so on. So once all this was done, I wanted to give you a little bit of an idea about the timeline, because I told you that when the patient presented, she was already 17 years old, and typically these patients die in their teens and so on. So it really needed efforts where all possible stakeholders from across the world actually came to support this. So we had Oren Devens's group. He's really a big shot in the field of neuropathologies, who gave us his reagents. We had only one single scientist in the world who studies this disease, because it's an ultra-rare disease, and she generated antibodies for it. She shipped them all within 72 hours to Delhi, and we started doing all these stem cell experiments, mouse experiments. Within 1.5 years, we finished all this, and then we approached our regulators in India, which is the DCGI, CDSCO, and we told them that, look, there is a patient who has to get this therapy. Can you help us? And they were extremely forthcoming. We had no idea that that so much of support is there for such kind of rare disease. And they said, look, this is a rarest of rare neuropathy. So there is something called a sunshine trial, or there is an N of 1 trial, where if a person needs a therapy to survive, then we would not ask you to go and do this in 10 monkeys or 20 apes or something like that. So you can go ahead. They gave the approvals, they audited the facility, and we started with the production of AAVs. The idea was that somewhere in 2024, so we also gathered a group of very heroic neurosurgeons who said that what would be the mechanism of doing this injection in the brain at Narayan Nethralaya, and figured all that out. But unfortunately, whatever we tried, in October 2024, this patient passed away. And in August, we had actually met this patient. And when we met this girl, she was already carried to this place with 2 helps. She could not see, she could not eat, she could not drink, she could not hear. She was in a very bad condition. Yet her parents were telling her that there are these doctors here who are trying to develop a new therapy for you, with that kind of a hope. So this talk today is actually in remembrance of Uditi. So Uditi is this girl whose parents actually put all the the hope in the basket of science. They wanted to fund and develop a new therapy all over by itself in a country like India, where there is no therapy currently existing in the world actually. And it required, almost, as I say, that it requires a village to do that. We also tried the same way, although unsuccessfully. But you can go and see this article that Nature has made. They had, they wrote about this whole process, which we tried to do in last year, to understand the depth of this. But it told us something very interesting, is that, Uditi is not isolated. There are many such patients from all over the country who have these kind of ultra-rare pathologies or rare diseases, for whom there is absolutely no hope. And that's the necessity to understand that there is a very long queue of patients, okay? And time is a commodity for many of them. Like in our field, in the clinical trials field, people continuously talk about safety, efficacy. And one of the prevalent thoughts is that you can make things safer and safer and safer. But at one point you have to give it to the patient to understand really whether it works or not. So you can do hundreds of mouse experiments, thousands of animal experiments, but actually the experiment that you need to do is to have possibility of giving these drugs to patients to understand, especially in the rare disease scenario, whether they are acting or not. And this is something that the FDA is also now slowly recognizing. So you can make these editors, and that's what we are trying to do through science. The pricing, I have told you, the moment we started developing these technologies completely, from the consumables to all the components in-house in India, the price dropped. So right now, we have, the cost, like I said, was $3 million US, which is around 20 CR. The cost that we could make right now is around 70 lakhs, close to what that is that of the CAR T therapy. But the government is not happy. They want us to make it much lower than that. because it has to be given almost free of cost to tribal patients, right? So the R&D keeps on moving towards those directions, more indigenization, more reduction of the costs associated with developing the pipeline, and developing on more technologies where we can scale it up instead of having to import from outside. IP and licensing.